A very warm welcome to everyone. I'm your host Bhavika Maheshwari and I represent Veeda Lifesciences. In today's webinar, we are going to talk about peptides in focus that are driving biosimilar experience. But before we dive deep into this session, let me quickly walk you through and introduce Veeda Lifesciences. As an integrated, tech-driven global CRO, we, Veeda Life Sciences, are committed to delivering excellence in everything we do. From preclinical to clinical trials and bioanalytical services, we provide comprehensive end-to-end solutions for innovator, biosimilar, and generic drug development programs for our global clientele. With sites across 26 geographies, Veeda Life Sciences today has become a trusted partner for our global clients. Together, we are catering to industries like pharmaceutical, biopharmaceutical, agrochemical, and industrial chemical industries, herbals, nutraceuticals, and medical devices industries. At the heart of everything we do is a simple principle: excellence in every step. It's what pushes us to keep innovating, to invest in advanced infrastructures, to collaborate strategically across the globe, and most importantly, to help turn scientific ideas into real world healthcare impact. So, as we begin today's session, I will quickly walk you through uh, the process. First, we'll begin with the presentation from our speakers. Then we'll move on to the Q&A sessions. The Ask a Question feature has been enabled. You can drop in all your questions related to the webinar there. I'll now introduce our speakers. Our first speaker for today is Dr. Prashant Deshmukh. Dr. Prashant is a senior research scientist at Veeda Life Sciences. He specializes in protein purification, biophysical assays, and biosimilar analytics. He holds a PhD from NIMHANS and has completed his postdoctoral research at NUJ Singapore. Over the years, he has led multiple GLP compliant method development and validation projects, particularly focusing on surface plasma resonance assays or SPR assays and BIA core based interaction studies. Our second speaker for today is Ms. Mehak Pandita. She is an immunology researcher at Veeda Life Sciences and brings over 4 years of experience in cell based assays, gene editing, and immunology development. Her work focuses on designing and executing immunology assays, including T cell and macrophage related studies and cytokine profiling. She assesses the immunogenicity of biosimilar drugs, and with a strong foundation in molecular and cell biology, Ms. Mehak plays a vital role in ensuring that our biosimilar programs are designed for success. Our third speaker for today is Ms. Keethana V. Ms. Keethana is a senior research scientist at Veeda Life Sciences. She brings over 5 years of experience in SPR based assays and biomolecular interaction studies. Her expertise spans across drug discovery, protein expression, and purification, and she has extensively worked with advanced biophysical instruments like BIA-Core 1S and the Octet SF3 SPR systems. She's passionate about translational research and she has contributed to several projects which is driving innovation in structural and biological sciences. So, without any further ado, I would like to call upon Dr. Prashant Deshmukh to begin the session. Dr. Prashant, over to you.
Thank you. Brief introduction for all speakers, Bhavika. And good evening, all friends. Today we are discussing here in webinar peptide bus based biosimilars. So title of this webinar is Peptide in Focus, Driving Biosimilar Excellence. Next, please. So peptides for therapeutic use. So I'm starting with the general definition of peptides. Peptide is defined as alpha amino acid polymers having 40 or lesser amino acid in their structure. And first, peptide was synthesized in way back in 1921, that is insulin, and till date, currently almost more than 80 peptides approved by uh, FDA for therapeutic uses, particularly different therapeutic areas like diabetes type 2, weight loss, irritable bowel syndrome, osteoporosis, prostate cancer, HIV, and chronic pain. Next, please. So, peptides for therapeutic use. Here in this slide, we are discussing peptides advantages and disadvantages over small molecules and biologics. So first coming over small molecules, like the peptides advantage and disadvantages over small molecules. So peptides confirms high specificity, good efficiency, good safety. Here high specificity is very important. I will give a small example of high specificity, why it is important. See, one of the small molecule, namely JQ1, it's one of the potential therapeutic molecule for nut carcinoma, and which is helping to reduce the cell proliferation in carcinoma, particularly nut carcinoma, and it is considered as BET family protein inhibitor, particularly BRD4 inhibitor. So this molecule JQ1 is inhibiting the cell proliferation via it is unacetylating the histone molecules. So thereby it's reducing the cells expression or cell proliferation. It's reducing the cell proliferation. At same time, this molecule BQ1, it's having like a toxic effect on different cells, which is required continuous their proliferation. For example, neuronal cells, then spermatogenesis. So thereby, specificity is important because JQ1 is having multiple targets here. So in case of peptide, this specificity is related to only mostly single targets. So that's why specificity is very important, thereby its efficacy and safety. And disadvantage of peptides lower its stability because of the molecules is consist of only 30 to 40 amino acids, and it is lacking secondary and structure stability. So that's why this molecule, in particularly in vivo, it is lower in stability. Its half-life is shortened, and most of up till 90% of peptide, it's having low permeability because it's working mostly with extracellular molecules, or it's binding extracellular receptor. So permeability of these peptides, it's less compared to small molecule. This is the disadvantage of peptide. When this peptide compared with biologics, its more important aspect is its low cost production. So peptide is production is required less cost compared to large molecule or biologics. And its other important advantage is low immunogenicity, and slightly compared to large molecules, it is having slight membrane permeability with the chemical modification. And disadvantage considered as low stability and short half-life. Next slide. In this slide, peptides for therapeutic use, landscape and global market, we'll just discuss. So, peptide global market expected to grow from current USD 40 billion to up till 100 USD billion by year 2034. So it's approximately CAGR per year and reaching to 11%. So if you see global market in past like a year 23 and 24, it's almost 38 billion and 42 billion USD respectively. Whereas when it compares to year 2034, it reaches around 117 billion USD. So it's global distribution of this peptides all over global. So in North America, it is almost like a 45%. Europe, it almost above the 20%. Asia Pacific region, it's around 18 to 20%. and Latin America it reaches to 5%, and MEA, it's like a Middle East Asia region, it's considering like a 2 to 3%. So it's a projected growth in like important countries, or most of country like this CAGR is around 3.2% in US, 3.3% in Germany. In France, it is 5.1, 3.7 in UK, in China, 8.7, and in India, it is reaching almost 10% by the end of 2034. Next slide, please. So peptides for therapeutic use, current research and future trends. Peptides offer unique opportunity for therapeutic modulation. In case of cardiac disease, ACE inhibitory peptides. This ACE is nothing but angiotensin uh, converting enzyme, and this angiotensin is responsible for increasing the hypertension. So this hypertension is the major disease in cardiovascular disease area. So this AC inhibitory peptides, other than AC, it's atrial natriuretic peptides, brain natriuretic peptides, C-type natriuretic peptides. All these NPs are very important in cardiovascular biology. Cancer P53 dependent alpha helical peptides, inhibitors of MDM2 and MDMX radiolabel peptides for cancers. In case of diabetes mellitus, it's GLP-1 receptor agonist. Intestinal diseases, especially antimicrobial peptides, that is called AMPs. These are playing pivotal roles to maintain the balance of epithelial cells as well as microflora. Then GLP-2 receptor agonists, for example, teduglutide. Then B, caspofungin for Crohn's disease. And one of the most hot topic is antiviral peptides. Because of these peptides are, as I mentioned, it is highly specific and highly effective. So this is one of the hot topic in this developing antiviral peptides. So all these clinical trials spread over multiple phases. Next slide. So landscape, global market, and future trends. So patent expiry for semaglutide in various part of world in 2026, it's getting expired in Canada, India, China, Brazil, Turkey, and more than 100 other countries. In the year 2031 and 2032, United States, Europe, and Japan. In case of liraglutide, in 2024, this molecule has been expired. But however, this generic liraglutide approved in US, Hikama, December in 24, Teva in August 2025, and EU, Aldao in June 2024, as well as Biocon December 24. So other molecule is tirzepatide. It's getting expired in 2027, and its core patent for molecule also getting expired in the year 2036. Next slide, please. So that's all from my end. My other colleagues, Kirtana, will discuss more detail about SPR binding assay. So she will take care of further slides. Thank you.
Thank you, Bhavika, for your warm introduction. And thank you, Dr. Prashant Deshmukh, for your insights on peptide biosimilar markets. And over the next few minutes, I'll walk you through how we approach peptide-based biosimilars from understanding its unique characteristics to developing an robust analytical method and demonstrating comparability between reference and test samples. I'll also touch upon key learnings from our recent studies and how they translate to a high quality outcome for clients. So let's begin with surface plasmon resonance based assays offered for peptides by Veeda Lifesciences. So in the initial slide, Dr. Prashant spoke about what are peptides and peptide drugs that are used in multiple therapeutic areas. So now let's see on GLP-1 peptides mechanism of action. So here, uh, if we see this mechanism, the GLP-1 peptide is a gut hormone that is released after food intake. So when this GLP-1 binds to GLP-1 receptor present in beta cells of pancreas, it increases the CAMP levels, which in turn stimulates the insulin release. And on the other hand, as the name suggests itself, the GLP-1 peptide, glucagon-like peptide, reduces insulin release uh, and slows down gastric emptying and reduces appetite. In surface plasma resonance assay, we see the binding of this GLP-1 analogs to GLP-1 receptor and measure the binding kinetics and affinity. And also with SPR, we can see the binding similarity of reference versus test drug products. Next slide, please. So in the current slide, let's see how SPR works and what does it measure. So SPR is a real-time label-free technique to study the biomolecular interaction, and it works on the principle of detecting change in refractive index near the sensor surface. So when an analyte binds to the ligand, there will be change in the reflected angle, and this change is proportional to the amount of binding. And as a result of binding, a sensogram is produced. So it is time against a response unit, and it gives us details on binding kinetics, which is the association and dissociation rate, and binding affinity, which is equilibrium dissociation constant, or KD value. So lower the KD value, greater is the strength of the binding. In an typical SPR assay for peptides, we immobilize this GLP-1 receptor in a suitable biosensor surface and pass the GLP-1 analogs, which is liraglutide, semaglutide, or tirzepatide over it as an analyte, and see the binding response. And in case required, we might introduce an regeneration solution to decay the binding complex formed. And then the KD values are estimated. Next slide, please. So why is it important to develop a method for SPR is because each biomolecular pair might behave differently. Hence, to obtain the reliable data, it is important to have a dedicated method. And there are several key factors to be considered when evaluating the method development. And before getting into it, when we say ligand in SPR terms, it is an interaction partner which is immobilized or attached to the ligand surface. And analyte will be the interaction partner that is passed over this immobilized ligand. So coming back to ligand immobilization strategy. So the ligand will be immobilized to suitable sensor surface, either be it by conventional coupling methods like conventional covalent coupling methods like amine coupling, or capture based for packed proteins. And once the immobilization strategy is optimized, we look into analyte concentration. So this analyte concentration is selected mostly around equilibrium dissociation constant, KD values. So like 10 times lesser the KD values to 10 times higher the KD values. And once the concentration is optimized, it will be injected either by using multi-cycle kinetics or single cycle kinetics method. And this analyte has to be passed from bulk solution to microfluidic system in certain flow rate. So otherwise, it might lead to mass transport limitation. And once the flow rate is optimized, when the binding interaction happens, the association and dissociation phase is seen in sensograms. So this association phase is also referred to as K on rate, and it should be long enough to meaningfully trace the association phase. Likewise, dissociation phase, which is also referred to as K off rate, and it should be long enough to observe the exponential decay. So once these conditions are optimized, as I said in the earlier slide, regeneration will be introduced if there is requirement to decay the complex formed. And apart from these key factors, there is one more important factor which governs the binding interaction studies, that is the selection of SPR running buffer. So this SPR running buffer should facilitate the binding interaction. So the components has to be selected wisely. So the component should maintain the pH of the interaction and with proper ionic strength and minimize the non-specific binding. And also, in case if we use any reagents that might lead to bulk refractive index, it should be eliminated with proper reference subtraction or solvent correction principles. And all these conditions are optimized to measure the true binding events. And once all of them are optimized, we have to fit the data with appropriate fitting models. So it can be the most conventionally used 1:1 binding interaction. So in case the data doesn't fit to this fitting model, other models can also be explored. Next slide, please. So once the method is developed, we have to validate the developed method to demonstrate that the analytical procedure is suitable for intended purpose. And for conducting this validation study, we will use suitably characterized reference materials. And there are certain parameters which is required to validate an SPR assay. So of that, the first one is repeatability and intermediate precision. So this checks how consistent the assay results are when repeated. So repeatability will be estimated on same day by same analyst, whereas intermediate precision will be estimated on different day by different analyst. So together they show that the method gives similar results even under slightly different condition. And then coming to specificity or selectivity. So in specificity, the ligand can detect the reference molecule correctly even when other substance like placebo are present. And in selectivity, it ensures that it does not respond to uh, unrelated or non-specific molecules. And then we have robustness. So robustness is tested to see how stable the method is when small deliberate changes are introduced. For example, we might introduce a change in biosensor lot and slightly vary pH conditions or buffer composition, etc. So if the results stay consistent, it means the developed method is robust. And then we see the system suitability. So it measures the overall assay performance. And by monitoring the immobilization level, chi-square, affinity, kinetics, and precision. So once all these parameters are evaluated, uh, so these parameters will also be evaluated with predefined acceptance criteria, which we will be seeing in the upcoming slides. And Only when the method consistently meets this criteria, it will be considered as validated and will be used for further sample analysis. And our validation approach is aligned with globally accepted regulatory frameworks such as ICH, FDA, EMA, and USB. Next slide, please. So here we are showing our in-house SPR kinetics and affinity data for tirzepatide. So on the left-hand side, we can see the binding of tirzepatide to GLP-1 receptor. And on the right side, we see binding of tirzepatide to GIP receptor. So both fitted using 1:1 binding model. And when we closely observe the binding pattern of the both receptors, there is a difference. Uh, and the KD values for GLP-1 receptors were around 40 nanomolar, which is high affinity. And for KD values of GIPR receptor is around 2.8 micromolar, which is slightly weak, but still significant. And this difference is because of dual agonist nature of tirzepatide when measured with both GLP-1 receptor and GIP receptor. Next slide, please. So as I said before, all the method which is validated will be compared with suitable acceptance criteria according to regulatory guidelines. Of that, the immobilization or capture level should be within 20 to 30% for each run. And the Rmax and repeatability of affinity and kinetic values will be within 15% of the CV. And whereas when it comes to intermediate precision, the values are slightly higher, which is less than 20% CV. And chi-square will be evaluated to see the goodness of the fit, and it should be less than or equal to 10% of the Rmax. Next slide, please. So when the assays is being developed, certain robustness parameters are already assessed. For example, like baseline stability, surface performance, and analyte replicates. But even in like for it will be revisited, verified, and documented at a later stage to establish and fully validated assay. And specificity, as we discussed earlier, it will not show any binding response to the target molecule, and it is tested with non-specific or closely related drug products. Next slide, please. So the sample analysis will be done after the method development and validation, and this also has certain acceptance criteria. Here, the KD reproducibility and chi-square is similar to the one in intermediate precision of method validation, which is less than 20% of CV for KD and less than or equal to 10% Rmax for the chi-square. And the test sample will be compared with the reference drug products, and the KD range will be estimated. So it is estimated using the formula which is highlighted here. KD range is equal to average KD plus or minus X times ESD. So here the multiplier X You can use an X multiplier to scientifically decide the suitable range within which the sample can be analyzed accurately. I would like to conclude with my presentation and thank you all for your attention. And I would like to hand over the session to Ms. Mehak Pandita, and she will take over and discuss about peptides in cell-based assays. Thank you.
Continuing from what Keertana just covered, we will be talking about the cell-based assays now, which are used in the peptide biosimilar development, and we'll discuss how they play a key role in assessing the functional similarities and immunogenicity. This image here shows us the mechanism of how blood glucose reduction in pancreatic cells happen. And this is, let's take an example of GLP-1 drugs. They are the peptide-based synthetic analogs of the natural GLP-1 peptide, which are chemically modified to enhance their stability and extended half-life. Now, when a biosimilar version of this GLP-1 agonist is developed, the key requirement will be the functional equivalence to the reference drug. So how it works is that GLP-1 analog binds to the GLP-1 receptor, which will activate the GS proteins. And this will in turn activate the AMP, which leads to the production of intracellular cyclic AMP. Now, this cyclic AMP does two things. First is it enhances the insulin secretion, and second, it inhibits the glucagon release. So now the levels of intracellular cyclic that are produced post the GLP-1 binding reflects how effectively the drug activates the receptor, and which then would be compared to the reference drug. Next slide. This is how a typical workflow of a cell-based assay for GLP-1 biosimilar will look like. First step is to choose a human overexpressing GLP-1 receptor cell line. In our case, we can choose HEC293 or CHOK1 cell lines. Now, second step is the seeding of cells, which will be done in a 96 well plate and incubating the cells with reference drug and the test drug product. Then the intracellular cyclic AMP will be estimated. Now, for estimation, there are various methods like alpha screen assay, um, which is a non-radioactive light-based method used to measure the intracellular cyclic AMP. How it works is it brings the donor and the acceptor bead close together, and when they interact, they emit light. So it works in a competitive fashion where free cyclic AMP competes with the labeled cyclic AMP for binding. So higher cyclic AMP levels will lead to the less light. To explain it in simpler terms, it would mean the signal strength will drop when the cyclic AMP increases, which allows you to quantitatively measure the receptor activation without using the radioactivity. The other methods, which include luminescence-based reporter assay and HTRF, which is a fluorescent-based method to measure the intracellular levels of cyclic AMP. The parameters that need to be considered while developing the assay are cell density, incubation time, edge effect, which basically means the variation which is caused by the evaporation and temperature difference across a 96 well plate. Next would be the concentration response curve or dose response curve, specificity, and sensitivity. Moving on to the slide. Now, this is innate immune response modulating impurities, which means they are the contaminants which can trigger the body's innate immune system. These impurities could either come from the product or the process, and they will trigger the innate immune system of our body. So, when they are They are present in very small amounts, and they are recognized by the pattern recognition receptors like TLRs. And once they are recognized, there will be cytokine release. And when the innate immune system is active, it can also lead to the activation of adaptive immune system. Now, process related impurities like it can come from the host cell protein or the growth media components or any other contaminants. And they stimulate the immune system by activating the antigen presenting cells. In case of related impurities, um, in our case, it would be peptide related impurity, which will trigger the adaptive immune system and activate the T cells. IRMIs can also impact the efficacy of the drug depending on their level and activity. If they are if the levels are high or they're very active, it will trigger the immune response and reduce the effectiveness of the drug. While in the low concentration, they can either there'll be very low effects or they'll be inactive, so they might not have any real impact. Next slide. The ANDA, which is abbreviated New Drug Application document provided by the FDA, reflects the current view on the criteria that needs to be considered while submitting for a biosimilar for ensuring the safety and efficacy of the drug. These impurities that are common should not go beyond the acceptable range in the in the reference drug. And for any new impurities, above 0.5% is not acceptable. And within the range of 0.1 and 0.5%, it should be adequately characterized before submission in an ANDA. Next slide. Innate immunogenicity is assessed in vitro by measuring the cytokine levels. It can also be done with complement activation by checking the complement activation using various reporter cell lines or PBMCs, which will help us to predict the potential immune activation. Now, in this ANDA document for the submission for a generic peptide, the FDA closely reviews the impurity profile as how it can differ from those in the recombinant DNA derived peptides and may trigger the immune system. Innate immunogenicity is broad and it's non-specific, so it will produce response each time, unlike the adaptive immunity, which is highly specific, antigen specific. And every with the new exposure, it will have a stronger impact. And activation of innate immune system will lead to the release of cytokines, chemokines, and acute phase proteins. Next slide. Yeah. As mentioned earlier, IIRMIs can activate the antigen presenting cells like macrophages and dendritic cells, which are decorated with pattern recognition receptors. And these cells will constantly monitor the surrounding through these PRRs, like TLRs, RIG1, and node-like receptors. And when they get triggered, if there is a foreign entity, they will activate the NF-kappa B and AP1 pathways, which will lead to the inflammation and immune cell recruitment. Now, if a protein is taken up at the same time, it can be presented with the MHC and co-stimulatory signals, which will activate the T cells. And if this activation is prolonged, your body will actually, start reacting to the proteins. Normally, it wouldn't. Next slide. So these are just the examples of PRRs based on their location. There are different kinds like cell surface PRRs, cytosolic, endosomal, and soluble PRRs. Next slide. Yeah. This poster from FDA titled in vitro immunogenicity assay for evaluating generic peptide drug product reflects their views on submission for ANDA. Now, there are several in vitro methods described in the literature for evaluating the immunogenicity risk of peptide products. And generally, these assays can be grouped on into two categories based on the immune response they generate. First is the in vitro assays for innate immune response. So this table right here shows the list of in vitro assays. Like for IIRMI assay, macrophage cell line like raw blue cells are used, and the readout would be to estimate the activation of NF-kappa B, which basically will tell you whether the innate immune pathways are being triggered. And likewise for innate immune response for stress-induced aggregates, human monocyte cell lines are used, and the readout here would be estimation of inflammatory cytokine levels. Next slide. Mm, This poster also outlines the recommended criteria for conducting the innate immunogenicity studies, such as testing the product both at release and at the end of the shelf life to confirm the consistent sensitivity. Other than that, we have also to show the highest concentration. We have to use the highest concentration that keeps the cell healthy. And for checking the signal strength recovery, we have to spike the product before testing. While using with I mean, sorry, using different cell lines with various PRRs is also a good way by which you can cover multiple innate pathways and different cytokine levels like IL-1, IL-8, IL-6 should be measured to indicate the presence of IIRMIs. Next slide. Now here on the left, we have the list of ligands that we have tested so far in our facility. And on the right side, we have the list of peptide biosimilars that we have worked on across different projects and clients. Next slide. Yeah, moving on to the in vitro assays to assess the adaptive immune response. And one of the recommended assays in the guidelines is in vitro PBMC derived T cell assay, which is done using the PBMCs from a healthy donor, and where the IFN gamma levels are assessed using ELISpot assay. Next slide. This is the pictorial representation of the workflow that spans about 16 days. And it is designed to expand the antigen specific T cells through repeated peptide exposure. And then IFN gamma levels are assessed using the ELISpot assay at the end of the experiment. Based on the FDA recommendation, it is also important to test at least 3 drug products, 3 reference batches, for any peptide impurities. Um, it also recommends that around 40 diverse donors should be used, which will help you to cover HLA and MHC variabilities. And controls for naive and memory T cell responses should be included. And it also recommends not to use the LPS, PHA, KLH for assessing the T cell response. Next slide. Right now, there aren't any standardized in vitro methods for um, or the assay procedures for assessing the immunogenicity risk. Different labs use various immune cell based assays, and some of them have shown a good correlation with the clinical immunogenicity rates, especially when they combine different immune cells, diverse HLA genotypes, and multiple assay readouts. But the key point here is even though these methods work to some extent, we still need to focus on optimizing the certain parameters, things like cell selection, assay conditions, control, and readout sensitivity. Really determine how reliable and predictive the results are going to be. Next slide. So at Veeda Life Sciences, to make sure we avoid any deficiencies in our innate immunogenicity study, we follow a very structured approach. We start with a custom cytokine panel and a well-defined study design that includes all the key parameters like cell number, cell variability, cell viability, and optimal culture conditions to keep the results consistent. We also include a proper control based on the assay suitability, and we monitor the signal strength recovery to make sure the system is working as expected. Uh, we also test different drug dilutions to assess sensitivity, and we also look at the drug formulations to rule out any matrix interference. And finally, we do the spiking studies with the known ligands to confirm assays responsiveness. So overall, this is the setup which will help us generate reliable and reproducible data and avoid any regulatory issues right from the start. Next slide. And finally, here is a quick look at the range of instruments available in our facility which support the assay development and analysis. So we have BIACore 1S Plus, Tecan Spark, Luminex, Agilent Novocyte, BDFAC, and Wenstop Robotics. Till now, we have completed more than 12 peptide studies. So we provide custom study design, comprehensive data package, and committed turnaround time with dedicated expertise. That would be all from my side. Over to you, Bhavika.
Thank you so much, Miss Mehek. Now we'll move on to the Q&A section, and we've received some interesting questions. So yeah, I'll start off with my first question. Uh, how does the mechanist nature of innate immune activation differ from that of the adaptive immune activation, particularly upon repeated exposure to the same antigenic stimulus? Miss Mehek, can you please answer that?
Yes, sure. So innate immune system reacts in a very general way, and it will react exactly every time it is exposed to a foreign entity. And but the adaptive immune system, on the other hand, it's more precise. So it actually learns and remembers the specific antigens. So if you're exposed to the same antigen once again, the response is much faster and stronger this time because of the immunological memory that we have.
Thank you so much. My next question is, what key experimental design consideration to minimize deficiency and ensure the robustness of in vitro innate immunogenicity assessment? Ms. Mehak, I'll ask you to answer the same question. Can you please?
Yeah, sure. So here at Veeda, we have a custom cytokine panel designed, which is specifically for the innate immunogenicity testing. And we have a solid study setup and clear reporting. The key considerations we have is that we make sure we focus on the cell number, cell viability, and appropriate cell culture conditions for the experiment, and we use the appropriate controls according to the study design. We also check the signal strength recovery. We run drug dilution studies to test the sensitivity, and for any formulation related differences, we have the matrix effect. Plus, we do the spiking studies as well for the known ligands to make sure that the system is working as expected.
Thank you so much, Ms. Mehak. My next question is, why is SPR considered a preferred analytical platform for peptide biosimilar testing?
So, as we discussed earlier, SPR provides real-time label-free measurements of binding kinetics, and this helps demonstrating a binding similarity between a reference and test products, which is critical measure of biosimilarity. And moreover, it's globally accepted by regulatory agencies for characterizing molecular interactions.
My next question is, how does SPR data support understanding of peptide mechanism of action such as for GLP-1 analogs or tirzepatide? Ms. Keerthana, can you please answer that?
Yeah, so SPR can differentiate binding profiles of peptides to multiple receptors. For example, tirzepatide shows measurable binding to both GLP-1 and GIPR, confirming its dual agonist behavior. So, differences in association and dissociation kinetics provide insights into receptor bias and functional potency, linking binding behavior directly to its mechanism of action.
Thank you so much. I'll ask one more question to you.
Yes,
How
please.
does how long does it take to complete biosimilar testing with SPR?
So if the scope includes method development, method validation, and sample analysis, it will take approximately 10 to 12 weeks, and it depends upon the project scope as well.
Okay, thank you. Thank you. What are the main challenges in developing peptide biosimilars? Dr. Prashant, can you please answer that?
Uh, Basically, this biosimilar development challenges are various different challenges we are facing, like general people are facing. Important one is achieving structural similarity in ensuring the biological activity. Upon that, um, like its quality, consistent maintenance of maintaining the quality of biosimilar. And key challenge, this biosimilar should be similar to marketed drug or RLDs. So these are the important challenges in peptide discovery.
Okay. I'll direct one more question to you, Dr. Prashant. How
Mhm.
do regulatory agencies view peptide biosimilars?
Actually, this regulatory agency requires strong analytical comparability data, as well as they're okay with like a limited clinical data if this biosimilarity assessed properly. And there are FDA approved PhD guidelines. In that, already mentioned how biosimilarity will get assessed.
Thank you, Dr. Prashant, Ms. Mehek, Ms. Keerthana, for answering most of our audience's question. I'd like to take a pause over here. We understand that this session has kept us engaged, but we won't be able to take any other questions. But please be rest assured, we'll mail all the questions to you. And also, we'll mail you a link of the recorded webinar. Can I go to the next slide? Lastly, on behalf of Veeda Life Sciences, I would like to sincerely thank all our audiences, our panelists, Dr. Prashant, Ms. Mehak, Ms. Keerthana, for giving us such wonderful insights. Thank you for giving us great learnings. And once again, thank you to the audiences for being such a wonderful listeners. We are earnestly looking to host you once again. Till then, take care, stay safe. Thank you, everyone.